Welcome everyone. I'm just going to take a second here to go over some of the logistics for today's event. We're going to get started with the content in a couple minutes here, but I'm just going to go over some information about continuing education and how you can participate today. So first off, we have a lot of great speakers lined up for you today. If you have any questions for the speakers, you should be able to type into the Q&A function of your Zoom bar. If you're looking at your screen, you should see that at the bottom. If you can see my cursor, go ahead and click that, type the name of your speaker, and go ahead and type the question. We will have some time at the end of every presentation to answer those questions. A moderator will prompt the question to the speaker. And make sure you don't add your questions into the chat feature. Chat is for comments, other discussion. We want to make sure that we capture the questions in Q&A so they don't get lost. But if you are using the chat, make sure you use the drop-down menu to all panelists and attendees so that everyone can see. For continuing education, we have California Department of Pesticide Regulation, DPR, and Certified Crop Advisor. For DPR, that's if you're a private applicator or a PCA or any of those, we have a half hour in laws and regulations, and then 2 hours in other. There's a course Coast Guard up for you. In order to get credit, there are a few things you need to do. First of all, there's going to be some poll questions that pop up on your screen. Make sure you answer those poll questions during the event. If for some reason you can't access the poll questions, or you are having trouble submitting, or it closes when you didn't finish, go ahead and just drop those answers into chat so we know that you're actively participating in the event. You have to be logged into the event for the duration. We are tracking that. So if for some reason you have to leave early, that's also okay, but you'll only get credit for the hours that you were physically present for the event. After the event, you're going to have to take an exam, which is those poll questions. They're just repeated on the exam. And a link will be emailed you before the end of today. So it won't come right away once the event ends, but by the end of today, by 5 p.m., you should get that in your email. You have 1 week to take that exam. And if you do not take it in that time frame, we cannot give you credit. So make sure you take that. You have to pass with a 70%. And then we'll get you your certificates and turn in your credit and all the good stuff. One last thing is if you're sitting in the office with a bunch of coworkers, only one person to device can get credit, so they need to log in on a separate computer or call in so that I can verify separate attendance and participation. For our Certified Crop Advisor, it's a little bit easier. At the end of the event, a QR code will be displayed, which if you have the CCA app, you can just open it up, take a picture, and you're logged in. If you don't have the app, no problem. You'll just email your name and your CCA number to ANR Program Support at ucanr.edu. Okay, and that's all I have. So I'm going to turn it over to Oleg to get us started for the day.
All right, thank you, Kelly, and thank you everybody who logged in so far. I hope we get a few more folks on this meeting. Appreciate the support from ANR Support Unit for helping us run this virtually. Hopefully we'll do this meeting in person soon again, as we did for the last 19 years or 18 years. So a 20th event. We'll have a kind of overview of production issues today. And it's only appropriate to begin this meeting with a presentation from our Agricultural Commissioner. And we're glad to have Ed Williams here with an update for what's pertinent to Ventura County, to our row crops. And Ed will take it over and discuss what he'd like to discuss with us today. Go ahead, Ed, please share and take it over.
All right, thank you, Oleg. Let me see if I can get this thing organized and start sharing my screen here. Let's see. Can you see the my presentation on your screen?
Yes, it looks great.
Yes.
All right, thank you. Great to be here. 20 years, that's great. That's amazing work that you guys have done, Oleg, organizing all of this. Thank you for putting this together. I really appreciate that. Today I wanted to go over a few issues, not a whole lot for you, and if you have any questions, please let me know in the question area. First, there's some new regulations that we want to cover. Decontamination is one that DPR has put into effect this year. A little bit of changes there, and we'll go over that. Paraquat status has been changed to a restricted material with some very specific restrictions that we'll try to go over. And then generally, I wanted to talk a little bit about fumigation and TIF tarps and some of those kind of things. And then the last slide I had to share with you are some of the non-compliances that we've run across over the course of the last year or so. I just want to emphasize that our goal is to make sure that we help growers comply with the regulations. We have less discretion nowadays, and there's a lot of pressure from the public to make sure that pesticides are done right. And so our goal in our office is to make sure that growers and applicators know what the rules are and how to comply with them. We want to make you successful in being able to farm in this country and in this area. So that's our goal, and we hope to help you do that. So decontamination, there's some new regulations starting winter of 2021. There are some non-ag requirements that mirror the agricultural requirement, and they're based on the American National Standards Institute standard Z358.1-2014 eyewash standard. So what is it specifically that the new eyewash standard requires? So at this point, there is a 10-second rule for use of the eyewash in the event of a exposure. It requires a 1/2 inch pipe, which I don't think they were quite as specific in the past of what size pipes and all those kind of things that are required, but now a 1/2 inch pipe is required. It also requires the use of tepid water. It can't be too cold, which can cause problems, and it can't be too hot, which can also cause problems. So it has to be there in the kind of that Goldilocks range to meet that middle tepid water standard. There has to be training provided on the use of the decontamination eyewash units, and it has to be tested on a weekly basis to make sure that it actually functions properly as you're going. It has to be able to apply 15 minutes or provide 15 minutes of water at 0.4 gallons per minute. And it has to be able to activate within 1 second. It has to be, and it has to be able to be activated hands-free. So just want to make sure that everybody understands that there are some new, very specific requirements for the eyewash equipment, especially that people have to comply with. Okay. So with that, I'll go ahead and put up the first question for you. The new eyewash stations, A, must carry 60 gallons of water. B, can only be operated after online course completed. C, cannot be too hot or too cold. D, must generate a pulse flow. Or E, require the annual testing. So please put the most correct answer on your test. And I think Kelly has the poll button up. All right, I'll go ahead and go to my next slide, which has the correct answer. The correct answer is C. It cannot be too hot or too cold. So it looks like 80% of the group got the question correct. So great. There is a minimum water flow requirement. There is training required, but it's weekly basis, and there is testing, excuse me, not weekly basis, but training is required. It doesn't require a pulse flow, and there is a testing, but it has to be on a weekly basis, not an annual basis. So, okay, so we'll go ahead and move on to our next slide and discuss the new Paraquat requirements. So there are new permit conditions that follow the new label. And for those people who have legacy material, they still have to follow the new requirements. So the handler now has to be certified. It cannot be somebody working under a certified applicator. It has to be the applicator themselves that's certified. There's an online course that's required. Hand or air application is prohibited. There's a minimum of 48-hour restricted entry interval. And then there's medium or coarser droplets that are required to prevent drift. And then no reduced personal protective equipment for closed system use. So we'll go to each one of these items and look at them a little bit. So this is a new restricted use material classification. It's due to acute toxicity, and it requires that the applicator be certified, again, not to be used by uncertified persons working under the supervision of a certified applicator. Okay. So there is an online course that is available that's required, how to safely use and handle Paraquat containing products and why it matters. Hand application or air application is now prohibited. There is a minimum of a 48 hours, so 2-day REI period. So nobody can go into the field within 48 hours. And it requires medium or coarser size droplets. Again, this is to prevent any threat or to reduce the threat of drift with this material. So it requires a little bit larger droplets for the application. And there is no reduced personal protective equipment, even if there is a closed system that's being used. So it requires full water impermeable protection for gloves and the mask and the respirator and the whole 9 yards. So there's no reduction for using a closed system. Okay. So the next question that we have related to Paraquat is handlers must be certified or applicators must be certified. B, they need to pass online course only if uncertified. C, can use less personal protective equipment if using a closed system. D, they can be uncertified if supervised by a certified person on site. And E, must file a worksite plan. So Kelly has the poll up for people to go ahead and answer. We'll give you some time to do that. Great, looks like you guys got this. For the most part, 87% chose must be certified, which is one of the new requirements in order to make the application. There is no online course for uncertified. Again, back to the first one, the applicator has to be certified. Obviously, cannot use less PPE. For a closed system, it requires full PPE and cannot be an uncertified person, even if that there is a certified person on site. So good job with that. We're winding up most of the presentation this morning, and we will have some time for questions, I think, if anyone has any. So the next thing I wanted to kind of cover, this is not new requirements, but it's something that we have some issues with on fumigations on an annual basis. And so we wanted to make sure that everybody is well aware of what the tarping requirements are for fumigations. If people choose to use totally impermeable film tarps, there is a 60% buffer zone reduction. So the distance between your site and the next site, next property site, is reduced by 60%. But the tarp itself has to be free from holes. What does free from holes mean? Actually, technically, it means free from holes. If you have nut grass that's poking holes in your tarps, you know, unfortunately, we have to stand by the standard, by the requirements, or you end up losing that 60% buffer zone reduction. So you know, 1 or 2 perforations from nut grass or something else, that can be fixed. It's not a problem. But you know, if we see a field, a tarp that's kind of perforated throughout, shredded, obviously we can't allow that to continue to be using the same buffer zones that are allowed for TIF tarps. So just make sure that you're paying attention to the tarps, that they're not being damaged excessively. Like I say, 1 or 2 holes, we're not going to get too picky with that, but just make sure that your tarps are in good condition, okay? And the tarps themselves must be marked by the manufacturers so that we know that it's TIF tarp. So it just gives you that credit. If we can't determine whether or not the tarp itself meets the specifications for totally impermeable film, we cannot give you credit for the buffer zone reduction. So just want to make sure that people are paying attention to those things. Apparently there's some problems right now with supply, obviously, for this and many other materials, but if you're purchasing from a second party, just make sure that it is actually marked, and we can give you credit for that, okay? Now, within a quarter of a mile of school, again, this is not a new requirement. This is something that continues, but these are our requirements. The application has to be completed 36 hours, at least 36 hours before the start of school. So in other words, you make the application before 6 p.m. on Saturday evening. That gives you 36 hours before 6 a.m. on Monday, when school generally people will start arriving at school and get ready for school the next week. So 36 hours application buffer zone there. And then there is an option that you can reach, that growers can reach with a school and with our office, the Agricultural Commissioner office, that permits a little bit of flexibility. We give a little bit of flexible alternative to some of this timing, as long as it's approved by the school and approved by my office. So that's in the regulation 6691F. And then it has to be, if there is an alternative that's reached, it has to be at least as protective as the 36-hour window before school starts. Okay. And the last slide I have for you, I just wanted to put up this is our top 10 list of total violations found. I believe this is within the last year. Food and Ag Code section 12973, conflict with label or the permit, just using material in conflict with the label. Code of Regulations section 6726, emergency medical information is either not posted or not available, which is a big problem. We take farm worker safety very, very seriously, and that's going to be one of our emphases to make sure that farm workers are protected, including personal protective equipment that may not be used properly or may not be, if the label requires water impermeable gloves, then we require that. Leather gloves aren't going to work. PCB not registered, pest control business is not registered with our office. We have several violations for that, and a lot of that is with some of our structural folks or landscaping folks. The decontamination, single-use towels or soap and those kind of things, you need to make sure that you keep up with those in the decontamination units that you have. No label at the use site, so the label is required to be on site when you're using material, and sometimes we're finding that there is no label available, and or there's no operator ID number available at the site. There's no record of training for farm workers or handlers that are handling material, which is another violation that we find. And then sometimes we're finding situations where there's a service container that has material in it, but it's not labeled properly. So we need to make sure that when you're transferring material to another site, that the service container has the label on it. And then the pest control business equipment is also required to be identified by the company that's using it. So those are the types of violations that we seem to be finding in the field. And again, our goal is to try to help you comply. As I said earlier, our discretion on whether or not to issue penalties has been much reduced by state legislation, state regulation, and so we're required to actually, enforce the regulations the way they're intended to go. But we want to make sure that you at least are aware of what to watch out for, how to avoid any problems. So I appreciate your being here today and the time that you're putting into this. So that's the end of my slides, and I may have a couple of minutes for questions if anyone has one.
Thank you so much, Ed. We have time for questions. Let's see if something shows up on Q&A box. Let's wait for a minute before we move on. Well, it looks like there isn't anything new. I think everybody who was participating did a really good job on the answers and they got information correctly. So that's great. And thank you, Ed, again, for stepping in and giving us an update on some of the topics for Ventura County agriculture.
Thank you, Oleg. So
if there's another question for Ed that comes to mind for somebody, please type them in chat and we can come back to this. But I'd like to move on and continue with the next topic, which is an update on strawberry breeding program with UC Davis. Steve Knapp wasn't able to join us today. He has a jury duty, but we're fortunate to have Glenn Cole, who works very closely with Steve and knows all the insides and outs of the breeding program, to discuss what's been happening in the breeding program, what are the progress and resistance development for some of the key pathogens that we have, and any related issues that he would like to discuss with us this morning. So with that, I'll turn it over to Glenn. And thank you again, Glenn, for stepping in and being able to talk to us this morning. Go ahead.
Thanks, Oleg. Yeah, happy to be here. Too bad Steve wasn't able to join. Looks like he's stuck in jury duty. So yeah, I wanted to give you an update on our breeding program, our academic lab, and the things that we're working on. And you know, as we all know, in the post-fumigant world, as far as tools that are available to address some of the soil-borne diseases that are problematic in the growing regions, you know, we've got less to work with. And so other tools that we have in our toolbox are genetic resistance. And so these are the things that we're working on to put into the products that come through the program, and also to learn more about the genetics and be on top of the diseases as they may change. We all live in a pandemic world, and we know that organisms change, and measures that need to be taken can be altered. So, nice picture of an organic field, and so, you know, we know farmers are moving to virgin ground. This happens to be virgin ground in Santa Maria, and as you know, the ground becomes used, it can become more problematic for soil-borne diseases. So, what's the program been doing since Steve and I started in 2015? This is kind of a culmination of our activity over the last 6 years. We've looked at 70,000 different hybrids, 1,800 populations, and from that work, we've identified and released 7 cultivars, and we've got 10 new cultivars that are under consideration for release, and we're ramping up to look at in a bigger way this coming season. In those commercial releases, that was as we went through the material that was left in the program from the previous breeding program breeders. We had 2 short day, 3-day neutral, and just this last year, 2 summer plant cultivars that were released. So lots of testing goes on to be able to make those releases. Yield testing, disease testing, and we want to bring forward products that are as strong as possible. And that strong disease resistance is a major portion of the releases as they come forward. We want to make sure that we're increasing our levels of resistance in the new products coming forward. For instance, we released 2 summer plant varieties. There was a lot of clamoring from certain growers that or end users that wanted a replacement to Portola because of sensory issues and or flavor complaints. Growers love it. It yields well, and it's Fusarium resistance. We released 2 products to address the flavor component, and so Finn and Mojo were released this past year, and very nice products, but they happen to be both susceptible to Fusarium, and so there's growers that have commented on that. So you know, we want to bring forward as best as possible products with the diseases that's necessary. So what are the main pathogens that we are looking at? 4 of them in particular, soil-borne diseases: Verticillium Wilt, Macrophamina, Fusarium Wilt, Phytophthora. Those are the key soil-borne diseases that have been shown to be most problematic for growers. In particular, the Fusarium has really sounded an alarm a little greater than the others. I mean, Macrophamina is also showing to be problematic. And so those are all things that we're focused on. We've identified that, you know, Fusarium Wilt is a gene for gene reaction, and we'll talk about what we're doing to address that. The other three diseases are all quantitative, which means it's harder. It takes more time and effort to incorporate the resistance genes that you're looking for, and then combining it all together is really the complexity of it all that makes it more difficult. So what do we have here? Looks like a pretty diseased field. Well, this just happens to be a field in Watsonville. That's at Albion that is severely impacted by Fusarium in this grower's field. And in the middle is Moxie. UCD Moxie was released in 2019 as a highly resistant, Fusarium resistant cultivar, a neutral cultivar. Again, this was all work that's been funded by the California Strawberry Commission. And we also have a USDA grant that we received in 2016, I believe. That is funding a lot of this disease work that we're doing. So Fusarium Wilt resistance we identified through the work of one of our PhD students, Dominique Pinco, that we've got a single gene resistance for Fusarium. Here you see a susceptible and a resistant cultivar next to it. So what's this allowed us to do? I mean, so we started this work in 2016. We screened a lot of germplasm. We threw out the net. We want to try to identify as much genetic resistance as possible. Along with that work, you know, our lab and our students and postdoc scientists, we mapped the strawberry genome, the octoploid genome, and in doing so, this has allowed us to use that as a tool to go and explore genetically germplasm to speed up and make our breeding more efficient. So in 2018, we identified this FW1 gene. This was part of Dominique's PhD thesis. And then that allowed me to go tap into that. And we developed a marker-assisted selections process where we identified the marker on chromosome 2, and we now can screen for those resistance genes and use that as a selective tool in our breeding program. And in doing so, 100% of the things that I bring forward carry that gene and are resistant. So we're using those genomic tools to assist the breeding program and bring forward better products. So what's that mean? It means speed breeding. So we've got these codominant Cas markers that we use, and because it's a dominant gene, we're able to bring forward products that have either in a heterozygous or homozygous resistant state selections that will confer the resistance that we need. So that really improves the things that I can then look at for all the other traits that we need to look at. I mean, there's a whole a litany of things that are important to strawberry besides just Fusarium resistance. And so we all know that it's got to yield, it's got to taste good, it's got to, you know, there's many things that come into what goes into a product. So, you know, our long-term strategy is to eliminate, you know, negative susceptible genes in our pool, and we're doing that quickly. You know, the DNA diagnostic tools are highly accurate, and you know, 3 of the recently released cultivars in 2019 all carry the FW1, and as I stated earlier, 100% of the things that I bring forward now have that gene. So with that work, we again cast a wide net so that we could look at all these genetics that have been developed over time. And we identified lots of sources of resistance to Fusarium. And so, you know, there's a lot of resistance that could be sitting around in other breeding programs, germplasm, and they can go develop, they can go use this tool. This information has been all publicly made available, and you know, other breeding programs can tap into it. I know there's been some reach out to Steve asking about that, and I think that, you know, some of these other breeding programs are using the tool so that everybody can be bringing forward Fusarium resistant products to the market, which is becoming more and more important. And we're seeing the problem up and down the state with Fusarium in the field, like that grower's field in Watsonville that I showed. So anyways, you know, we found that 2/3 of the non-UCD germplasm had resistance genes. And so we've been able to identify lots of sources. That's the key. And both from wild germplasm and from all old cultivars. You know, in the UC germplasm, there was about 40% resistance. So there was a lot of material that didn't have the gene. And, you know, and for years, the breeding program didn't need to look at disease resistance as a major focus, as there were other tools like methyl bromide that mitigated that problem. But now we're spending more and more effort on the genetic component. So that's FW1. We also identified 4 other Fusarium genes, FW2, 3, 4, and 5. 2 of those also occur on chromosome 2, and 2 of the other ones occur on other chromosomes. Well, why is this important? Again, these are other genes. We know the sources of these germplasms. We can stack these genes into our FW1 germplasm if necessary, because you need to have backup strategies for the potential for race changes or you know, the disease changing and evolving. And so, you know, we've got this germplasm, we know the sources, we may need to tap into it in the future. We are developing markers for these so that we could, again, do this genetically, and it makes the process much more efficient and effective. Again, this was work that Dominique Panko did in conjunction to her PhD. So, and along those lines, we know that there are Fusarium races that occur in other parts of the world. You know, our work has been done with California race, but we wanted to again broaden that research look out to what other races there are around the world. And we know there's a Japanese race, Australian race, and a Spanish race. And so we wanted to assess germplasm, identify resistance candidates. And so in that work, and again, this work for these other races wasn't done out in the field, don't worry. It was done in very controlled environments here on campus, in growth chambers, where the pathogen was kept under control. That work was done with Peter Henry, who was a research student in the pathology department here, has since moved on and is now working at the USDA in Salinas, and is a scientist who we collaborate with. And then also Dominique Panko worked with Peter Henry on that work. So we've identified this. We know the resistance selections of these other pathogens. And again, it's another set of things that we've got that we're prepared for in case these other pathogens evolve into similar type races. So we have outstanding short day and neutral cultivars with Fusarium resistance, and Victor, Warrior, and Moxie were the ones that were released in 2019, and summer plant varieties did not have Fusarium resistance, but we're addressing that with the new releases that are coming out. So let's move on to another story, which again, Macrophamina, which is an issue in the Ventura region, can be a significant issue. And again, Macrophamina is, well, what happened here? Macrophamina is something that comes on because of a stressful environment, whether it's water stress or heat stress. It brings on the disease. The Macrophamina is pretty ubiquitous. You find it in a lot of environments, and so it's going to be a problem, and has shown to be in many areas. I saw some areas in Ventura last year where the disease was a problem. So that was something also we, as we began our breeding efforts, we were looking at all these diseases and again, casting a wide net to identify resistance genes. So what did we do in 2016? We're looking here at, we looked at 576 accessions in 2016. Well, out of those, only 2% were resistant and 98% were susceptible. We did a wider set in 2017, and we pretty much saw the same thing. So the only resistance things we saw, the 1s and 2s down here, are levels of resistance were in the 2% range. Well, that's pretty indicative of Macrophamina. I worked in other crops where Macrophamina is similarly very devastating. It's usually very low levels of resistance. So can be a devastating disease in stressful environments. So we wanted to attack that and go after, improvement for strawberry. So we identified those resistant things, and I put together and developed some elite donors that we had in our program that had some levels of tolerance, and we crossed those into things that were highly resistant. And we created 16 families to test. So in 2019, we did that testing. And there were roughly 2,200 individuals in the field. And this is what we saw, which was a surprise. We didn't expect to see this level of resistance. So here we see resistance, the 1s and 2s are levels of resistance. And so over 50% were highly resistant individuals from those 16 elite families. Out of that, we identified 23 individuals I selected to intermate with each other to combine those resistant genetics. Again, typically Macrophamina is considered to be a complex quantitative trait. So cycle 2, we made those crosses in 2020, and here in 2021, we tested 1,152 hybrid individuals, and our resistance levels were increased even higher to 76%. And so this was really quite fabulous. And I'll show you some pictures, because pictures always are worth 1,000 words. So this was our 2019 field. These were all families that had resistant by resistant crosses. Well, you can see that these sets of families in here were uber highly resistant. We had these were, and again, we test in a very extreme environment here in Davis, where it gets very hot, and we regulate the water so that we can create the stresses that we need. And so, but we selected things out of all these families, even some of these families where they eventually died. This would be later in July. There were selections here that, like that one there, that were flagged and were resistant. And we intermated a lot of these to create the families that we tested in this past year. So this is the look of this past year. And this is the 76% that were resistant. It's basically, this was again in July. We would anticipate seeing a lot of death in here. And we have some checks in here that were dead, but basically it's a carpet. And so this was really very encouraging We continue this work, and we are intermating these new materials coming out of this field, and we're taking selections out of this to put into test. Because, you know, finding genetic disease resistance is important, but again, they got to do all these other things like yield and taste good and the whole 9 yards. And so we're at the process of bringing these materials forward and testing those. So lastly, I'll just end on this slide here, I guess, is we're also working on Verticillium Wilt. Again, another quantitatively inherited disease, a little more difficult. We anticipate, you know, using genomic prediction tools to help us improve, which will work for both Macrophamena and Verticillium Wilt. And in Verticillium Wilt, we are doing that with some populations this year. So where we can identify, you know, with using the genotype, identify those selections that have a higher probability of being resistant, and taking those selections to the field to look for yield and fruit quality and all the other things that we look for. And so again, it makes we can do that at screening level, and when it's a seedling before we put it in the field, and we say, okay, I've got 2,000 things here, maybe 10% is good, and it's in this little zone down here. I can take those 10%, put them in the field. Again, it saves space in the field. It increases the probability that we're going to have something that we can pull out of that. And again, we're making sure that they all have Fusarium resistance. So I've got a chunk of stuff that's in the field that's from, you know, this look here, it's this genomic look that's got Fusarium resistance, and again, so we can come forward with better products. So what's the take home on this? Genetic resistance is important. It's what we can do from a breeding standpoint to help the industry bring products that are more resistant, as the other scientists are working on other tools to help the farmer address these needs, and especially in an organic world where there aren't soil fumigants being used. And so those are the things that we can do to address the issues. So I think that pretty much takes up my time. Oh, I guess we did have questions. I wasn't part of this, so I don't know. Should I ask those questions now?
Yeah, so you should see this poll questions. There's 2, so make sure you scroll down and answer both. You can read them, Glenn, just go ahead and read them.
Yeah,
and
sure.
Then by the time that you finish reading them, I think it'll be time to go over the answers. And if you
Sure.
don't know the answers, I have them written down somewhere, but.
No, I think I can figure these out.
Okay.
Can natural genetic resistance to Fusarium Wilt prevent production losses? No, yes, but the resistance is weak. And 2, does natural genetic resistance to Macrophamena exist in strawberry? Yes, but resistance is genetically complex and complete immunity is lacking for this disease, or no? Very good. Can natural genetic resistance to Fusarium Wilt prevent production losses? The answer is yes. I see there's a large component of respondents that say the resistance is weak. We have not found that to be the case. And we test in very extreme environments. And so, and then does natural genetic resistance to Macrophamena exist in strawberry? And the answer is yes, but the genetics are, resistance is complex. So most of the respondents got that correct.
Great, thank you.
Thank you, Glenn, for stepping in, discussing this with us. There should be no questions. If there's questions comes to mind for Glenn, please type them and maybe we can come back to this. We'll move on with the product. Yeah, as Glenn mentioned, resistance it's pretty key for us, and something we noticed back in the days when we had Macrophamena and Fusarium already discovered. And the first time when we did those trials with cultivars like Camarosa and Ventana and Camino Real, we knew there's resistance, and it's excellent to see that both UC and other breeding programs are focusing on using the same tools and identifying resistance to those key pathogens for our production. In fact, this is just a short list of what we have to deal with. In fact, we can't control those pathogens with the fumigation tools that we have today. So we have to live with them. And as we live with them, we have to rely obviously on resistance, but all the other things that we can consider in this kind of IPM approach. And one of the key ones is preventing plant stress, being able to grow plants when the conditions for development of disease are really slow, or disease doesn't develop fast, such as growing, producing fruit in the winter, as we do in Oxnard. I think that's pretty much one of the keys. But as these programs move forward, it's really tempting to evaluate every cultivar, every selection for tolerance and resistance. But it's not possible. But we have some questions we'd like to answer as we look at those new cultivars coming down the pipeline. How much pathogen in soil can strawberry handle if you have susceptible cultivar? Can it grow at a really high level of the pathogen in soil, in very infested soil? On the other hand, if we have cultivars with either moderately tolerant or susceptible, can they do okay if the levels of pathogens is low? So I'll summarize a Fusarium data from the last couple of seasons, and then this year's Macrophamena Fusarium test that we had conducted and continue to do so this season as well. Now, how do we do this? I would like to do it all in the field conditions. We have a fumigated clean soil with planting holes cut out. We'll excavate the soil from planting holes and create what we call an inoculated environment with different pathogen densities mixed into that volume of soil, whether it's a Fusarium or Macrophamena. It's mixed and placed back in the planting hole. So when the transplant comes in, the roots that come out of the crown and new roots coming out from the old roots immediately come in contact with infested soil. And we looked at the levels from 0, we call them from 0 to 4, with increased population of coliform forming units per gram of soil. So concentration of pathogen increases from 0 to 4 in all those root zone soil volumes. And we started with free cultivars. They all planted in replicated plots. And the first time we started this project was with Victor, Albion, and Monterey. The idea was to look at something that's newly developed for Fusarium resistance like Victor, Albion, which is we know very susceptible, and Monterey, which is moderately susceptible, moderately resistant, depending how you like to call it. And a couple of things that came around from that work. This is fruit yield. Again, all those charts will have 0 on the left, which in clean soil with no pathogen, and 4 is the highest level of pathogen inoculum. That cultivars such as Victor with good resistance, they have pretty much the same yields in clean soil at 0 as the highest population of Fusarium in the root zone at 4. And for the other cultivars, you don't see a whole lot of things going on. They're holding pretty steady, even with the known susceptibility, until we get to this very high level of pathogen presence in soil, level 4, then the yields drop off dramatically. And this is just a visual. Albion susceptible at 0, clean soil does well. Level 1, some low level of pathogen, occasionally we'll see some crowns dying, and the same with level 2. As you go to level 3, concentration in the root zone increases. We have diebacks with full crowns, half crowns. It becomes pretty prolific in decline. So as expected, mortality rates in those cultivars, this is end of experiment in end of June, July. One of the important developments here at all levels is a blue line on the bottom that stands for Victor. There's no mortality regardless how much pathogen is in the soil, in the root zone. Then there's what we would call moderate susceptibility or mortality in Monterey, the brown line. And of course, Albion being very susceptible, it's almost a straight line going up. You get 100% mortality already at level 3. So it makes a difference what you plant and how they react to it. We looked at some of the relationship. The important part, there's no relationship between Victor and Fusarium inoculum levels. So regardless of what's in soil, the yields seem to be the same. For Albion, the relationship was pretty weak. As we noticed, 2 all that high rates of Fusarium that we saw as yield losses. For Monterey, also a relatively weak relationship, but there is one. As you see here, as you increase population in the root zone, the total yields of Monterey go down. Mortality, that's how we lose plants. Again, no relationship between inoculum density and mortality for Victor. So it looks like it's resistant both in terms of mortality and in terms of yield at all levels that we tested. Albion, this is that straight line we expected. This is almost a perfect fit. The more pathogens in the soil, the sooner you're going to get, the more plants are going to die in case of Albion. There's might be a little bit of a lag between zero clean soil and very low level of 1, but as soon as you get higher levels of pathogen, it goes really fast to 100%. And Monterey, there's a kind of moderate slope for this dotted line, which is a modeled line for the mortality. That could give you some indication or projection of what to expect based on your soil sample. If you have known pathogen density, what to expect with this cultivar. And at the end of the season, we wanted to confirm that things were as we expected. So on those two charts for Albion and Monterey, you'll see mortality given in bars and concentration of coliform forming units per gram of soil in those planting holes and then the season, and they pretty much correspond. The higher concentration of the pathogen at the end of the season, the higher mortality. So as expected. We also looked in 1920 season at short day cultivars, Petaluma, Victor, and Warrior. Petaluma being susceptible to Fusarium, and as we saw previously, Victor and Warrior being resistant. They grew about the same rate. In fact, Petaluma grew a little faster in December. Now going to be a series of plants that look relatively healthy in March and April. This is a picture of end of April. There's no signs of disease. You could look at Victor. All the plants look about the same at all levels of pathogen. And the same goes for Warrior. Now, if you look at the yields January for April, the lines are relatively straight. There's not much going on. Petaluma yields a bit less than other cultivars. But other than that, there's not much else to say about this. When we look at the yields for the whole season, we see that Petaluma lagged behind about, and by the level 4, we got about 36% yield loss in Petaluma. It didn't seem to be dramatic, but it did accumulate as you move to higher concentration of pathogen. Percent mortality, again, Victor looked pretty good. There was some mortality in Warrior, but Petaluma was the one with the most mortality, reaching about 40-45% in July. What about Macrophamena? We completed the project this season. Same approach, 4 levels from 0 to really high level. And again, if you look at plant size, there's no effect on plant vigor. The plants grew at about the same pace early in the season. If you look at fruit yields, all the way from March, you didn't see much going on. We know Macrophamena is stress pathogen, comes in the warm soils late in the season. If you look at January through March, the fruit yields look fine. Yes, Petaluma yields a little bit less, but overall there's not much going on. The plants look fine. This is in March, good production, no signs of infection. However, if you look for the whole season, especially May and June, beginning of July, we see those dramatic drops in yield due to plant mortality. And that was true for all cultivars we tested, whether it was Victor, Warrior, Petaluma. They all drop yield pretty rapidly. I think one of the maybe noticeable things that with the yellow line standing for Victor, at low levels of Macrophamena, we didn't see significant drop in yield. But as we progressed with a high density of population of Macrophamena in soil, they all followed the same fate. And of course, Petaluma, as expected, was the worst. We have nice fruit in March, good production again, as I show here. And then this is a situation in May. Petaluma, Victor, Warrior on the left at low level, second lowest level. There's some mortality going on already in all 3 of them. And at highest level, level 4, on the right, you see pretty much complete death. Well, percent mortality, again, not much to see until April. There's some mortality picking up at high level of pathogen and warrior and petaluma. But as soon as you get to May and June, as expected with warm soils, our level of mortality go almost linearly all the way to high level 4. So in short summary on that topic, vector and water resistance at high pathogen density was pretty excellent. And I think that's true for many other cultivars bred with the same technique as Glenn described. They've been shared with other breeding programs. We have good resistance. And I think it may be applicable to other cultivars in that spectrum. For susceptible cultivars such as Albion, Monterey, Petaluma, and some others, it seems like we can produce a crop while Fusarium infestations in soil is low. I'd argue perhaps that's we've seen those levels early before we discovered the pathogens themselves. Maybe occasional dieback or decline was just misdiagnosed and mislabeled. So we might have been having those issues before we even had serious losses just because they've been masked by ability of those cultivars to produce at low levels of pathogen. And as we increase the pathogen density in soil, which does happen rapidly, we see those massive collapses that we've seen in many slides in this talk and before. Macrophamena, still to be a tough test. It's good to see a promising germplasm coming around with the breeding programs. As we saw in the Vector, it might be okay to tolerate low Macrophamena density in soil, but as soon as it goes up, it was very susceptible. We'll be looking for Anteras, our key cultivar here. We know it's susceptible. We'd like to quantify it. We'll also look at Mojo coming with the UCD breeding program as well. See what it does against Macrophamena in the field conditions in Ventura. Just one point to end this talk is it's great to have those traits that help us dramatically, but all cultivars support pathogens. The point I make is from work from Peter Henry and his colleagues that showed all the bars in this chart, whether it was susceptible cultivar, resistant, support population of Fusarium oxysporum form specialis fragaria. The pathogen can multiply on strawberry roots, on raspberry roots, on the roots of other hosts, well, and persist in your soil. So having a resistant cultivar looks really great, but it doesn't resolve your Fusarium problem. I'd like to acknowledge several parties that helped with this work: the nurseries, the Hansen Rec and staff, and NIFA grant funding for this. And before I move on to questions, I'd like to finish up with that second part of my presentation about nutsedge, which will take just a second. So, that's one of the world's top 10 weeds that we've been very successful growing here in Ventura County, and it's also weed in Santa Maria and some interior valleys of Salinas and King City. It's a troublesome one, and we have fewer and fewer tools every year to control it. It can be really devastating, especially once we moved away from methylbromide, methyl iodide fumigation. Those were historically the best tools to control nutsedge. We lost some herbicides in process as well. For example, with Lotsus. The question was, can we make S-metolachlor dual magnum work for nutsedge control? It's not we knew for a long time it's a good material against yellow nutsedge and maybe purple nutsedge. Used in vegetable crops, we use it in strawberry under HDP and low density polyethylene film as well. The question came around is, can it be safe and efficacious under TIF, both in summer and fall planted strawberry? So just a background of this, we worked with this since 2009, maybe even earlier. And the way we looked at those products and rates is we like to have a pathogen, in this case, tubers of the weed and nutsedge placed in pots so we can have same physiological age of the weed propagules. And we started by looking at the high rate, 0.95 pounds of active ingredient per acre, applied to beds covered by mulch. The idea is to apply it just like we do with like oxyfluorophane or flumioxazin as a bed applied material, because that's we're going to see most of the nutsedge damage coming from the plastic. I will look at both purple nutsedge and yellow nutsedge as well. We almost always have yellow nutsedge here as a problem species for us. Well, treated check got weedy pretty fast, and we had pretty good control with dual magnum at this high rate. And this is purple nutsedge. The red line is dual magnum high rate, and white is the number of shoots in untreated. So we're doing pretty good. It was even better for yellow nutsedge, the one we're mostly interested in. This is a white line going up with germination of shoots, and the red line looks good. So we knew the high rate would work well. The important part was to look at quality of the strawberry plants if they've been affected by this herbicide. And so what was interesting, you also looked at the spring cohort, and there was some residual nutsedge germinating still in the treated check and hardly anything in the dual magnum at high rate. So in 2010, we looked at a lower rate too, and we looked at how do the plants respond to this S-metolachlor. And you couldn't really see much here on those pictures. They might look a little smaller. And you look at injury score, which was scoring from 1 being no injury to 10 being dead. There was no significant differences. But then we look at marketable yield, and we see this response on the right. By May, the plants have been behind in production, especially at the high rate 0.95. We saw significant yield losses. Now, this is a kind of herbicide that doesn't cause leaf burn or twisting or severe spotting. It slows down the production of roots and growth. So it's kind of a growth inhibiting herbicide if to susceptible species. And so the plant just lagged behind enough to have those yield losses. We looked at lower rates, which was, I think, a good direction to go. And they were still efficacious as a check on the left and recheck on the left. And all the rates that we looked at seemed fine. There was some survivorship in 0.33, but I'm pretty satisfied with the control levels under a regular film. And injury was not significant. And the yields were numerically maybe lower, but not statistically lower than the check, which you see on the left. So in 2014, there was a intent to have the label. The product has a federal label, and so it can be used. And the hope with it, we're working with Syngenta, can develop a rate applicable to California, can become part of one of the labels that we can use here in Oxnard and Santa Maria. And the use was 30 days pre-plant, just like other herbicides. We know it can be tank mixed with Gol or Chateau and be efficacious as well. So this year we looked at summer planting and fall planting as well. We looked at those lower rates, zero being the check, 0.33, 0.66 pounds AI per acre. And it was applied 35 days before transplanting. And of course, we wanted to look at crop performance and injury. That field had very little to no nutsedge, so we couldn't make a justification about the weed control. But here's a chart showing the plant sizes. This is under TIF, so this is a new modern approach of trying to make it fit. The line with black circles is a check. We see that the low rate in a plant growth, and the plants grow pretty fast, nearly equals the progression of growth in the check. So they know they're not staying behind. High rate, we see that the rate of growth is slowed down by 6 weeks after planting. This is an injury score, again, from 1 to 10. We don't see severe injury. We see just very slight stunting. But if you follow those lines from first week into the 6th week, you could see that, again, the low rate is somewhere intermediate between clean and high rate, the untreated soil. And the high rate is 0.66. We see that the injury scores, those triangles here, are the highest. So it looks like we have some rate response. I hope we would have significant yield losses at low rate. We continue to look at fruit production. Here's some visuals of this. Untreated check. Those plants are growing rapidly. Very little difference from low rate 0.33 on the right. But if you look at 0.66, the plants don't fill up the space quite as fast. They're just a little bit behind. This is another way to look at it. The yellow line separates 0.66, the high rate of dual magnum versus untreated check in front. Those plants probably yield a bit less early. Maybe they will catch up. We'll see. It's important to know that. We're also looking at this in fall planting. We'll see how the nutsedge control, in this case, we bury the tubers in beds and we'll evaluate them for efficacy. But we'll look at how those winter planted fronteras will respond to dual magnum rates. Looking also at light and heavy soils. The hope is to find that niche, the golden middle, where we can still have good efficacy without plant injury. And that's where that project is at. And I think I'm out of time. If there's a moment, I could take a question or two.
It looks like there's one question
Okay.
in Q&A. Can you open it up?
Yes. Nutrient mixes, conventional organic, affect the results of the experiment. What is the grower standard nutrient mix that's used in different cultivars in the experiment you presented? Very briefly, we don't do anything differently. Is about 300 pounds of triple 16 fertilizer put out. So it's a conventional system. We don't really do anything differently for those treatments. So they're all uniform and considered as a standard. So we don't really pay too much attention to that part.
Okay, so there's also a poll. I just launched it. If there are 4 questions, so please, everyone scroll down, answer all 4 questions. While people are working, we can get the next speaker set up to get ready to go to present. And then Oleg, I'll ask you to go over the questions briefly.
Yes, so the question was about Fusarium density in the root zone increasing mortality equally among all tested cultivars. It looks like a split decision. The reality is that it was not equal. Some cultivars did not respond to pathogen density, so the no answer is correct. Does planting and higher resistant strawberry cultivars decrease Fusarium in soil? The question was answered mostly correctly, 59%. Actually, let me just back up. Resistant cultivars do not provide control of Fusarium. So the correct answer is no, because the pathogen can multiply on the roots of any cultivar. Third, dual magnum was methyl cause strawberry mortality. The correct answer is no, and most everybody got it correctly. And the final one was the rate response to dual magnum in strawberry. Correct answer was yes. I think everybody got it correctly.
Great, thank you. We can move on to the next speaker.
Yes, and in the theme of pest management, we're going to hear update in our Drosophila research. In strawberry, it's obviously a big issue, both conventional, especially organic production. So with this, I'm going to open the floor and pass it over to Fatima and Frank Zalem, who worked in the lab and in the field to conduct their research studies. And with that, please go ahead.
Thanks, Oleg. Can you see my right screen?
Yes.
Yes, it looks good.
Okay, thank you. So yeah, I'm happy to give you some update on Drosophila research we've been doing in strawberries in the last couple of years. So spotted wing Drosophila is an invasive vinegar fly, which is from Southeast Asia. It was first reported from strawberries in Santa Cruz County in 2008, and since then it's been a serious pest of berries and also cherries here. Both fresh market and processing strawberries are susceptible to spotted wing Drosophila. In fresh market strawberries, some cultural practices and insecticide applications can help protect fruit from this vinegar fly. In processing strawberries, because there are longer intervals between harvest and fruit remain in the field for a longer period, they tend to become softer and more susceptible. And that's the time that insecticides applications stop. So the problem is worse. And also insecticides mainly affect adult flies, and they're less effective against the larvae that are inside of fruit. So to reduce the population, the options are to have shorter harvest intervals and also to more frequently remove the overripe fruit from the field. When we started this research, our objectives were to assess what's the Drosophila larval infestation in both fresh market and processing fruits, which we call ripe and overripe, from three main strawberry growing regions in California. We also wanted to know what's the proportion of spotted wing Drosophila to other species in both fruit stages, and also assessing what's the extent of resistance in spotted wing Drosophila populations to different insecticide classes that are commonly used in strawberries. For that purpose, we collected fruit, both ripe and overripe. And as you can see in the picture, the ripe is more firm and solid fruit, and the overripe is the fruit with softer skin. With the help of our collaborators, we collected fruit from 2 to 4 sites per region, and in 2 years, mostly in 2019 and 2020. So when we got the samples from half of those samples, we collected larvae, and this is how we did it. We gently crushed the fruit and then floated them in a water and salt solution, and then strained with fine strainers and collect the larvae under microscope. Then after counting the numbers, we also gave a subset of those samples to Dr. Joanna Chu at UC Davis. She has developed a technique that uses molecular markers to discriminate spotted wing Drosophila from other Drosophila species. So we were able to determine the proportion of spotted wing Drosophila in our samples. From the other half of fruit, we reared flies, and we kept the fruit in plastic containers on a layer of sand and cotton, and then kept them in growth chambers for a period of 2 weeks. And once the flies emerged, we used morphological characteristics to identify them to species. So for the females, we looked at the ovipositor, which should be serrated in spotted wing Drosophila, and also the dark spots on wings and front legs in the males. So all spotted wing Drosophila flies were moved to these diet bottles, and we started colonies to do insecticide work with them later. So here is what we got from our Oxnard collection. So in 2019, results were consistent with our general hypothesis that there will be more Drosophila flies in overripe than in ripe fruit, and also more non-SWD in the overripe fruit, which has softer skin. In some cases, like in the site 1, when we saw more non-SWD in the ripe fruit, we believe that it's possible that spotted wing Drosophila first laid eggs in those fruit, and once the skin become soft enough for the other species, they invaded the fruit. We were not able to collect any fruit from Oxnard in 2020 because of the pandemic, but in 2021, we got fruit, but those fruit were not were overripe and very overripe. So it was just hard to discriminate between different ripeness stages and to explain the data at this collection. But at least we could start colonies with those flies and do insecticide bioassays with them. From Santa Maria in 2019, we collected flies from 3 sites. These were fall planted berries that actually, the end of the August is actually the end of the season for this berries is the beginning of August, and that's when we collected those strawberries. So that's late in the season for those flies. But the S4 is summer planted, and when we collected was early in the season. And if you look at the y-axis, you can see that the infestation was very low. So we just know that early in the season, the infestation levels are very low in strawberries. And then in 2020, we collected samples from 2 sites. Again, the infestation levels were so low that the difference between SWD and non-SWD flies doesn't really show. In Watsonville in 2019, we collected samples from 3 sites and twice per season. The numbers between ripe and overripe fruit were not different, but the infestation was much higher than what we had in Oxnard and Santa Maria. And then more than 89% of flies that were collected in first collection in the ripe fruit were spotted wing Drosophila. And in second collection, 100% of those flies in the ripe fruit were spotted wing. We did the same thing in 2020, had 2 collections from 4 sites. Again, the number of flies, the average number was higher in overripe fruit than in the ripe. And almost 100% of flies in ripe fruit in first collection were spotted wing Drosophila, which is consistent with our hypothesis that spotted wing is the first to attack the ripe fruit, and then once it becomes softer, the other Drosophila species can come and successfully oviposit in the fruit. For detecting insecticide resistance, we used a technique that has been developed by our collaborators. It's called rapid bioassay, and it uses this discriminating doses that have been established based on the concentrations that are needed to kill 90% of a susceptible population times 8. And that is a susceptible population that was collected from an untreated blueberry orchard in Michigan. In California, we have also a susceptible population from winters. And for spinosad, we use the concentration that kills 99% of that population times 2. The method is that we use these scintillation vials and pipette 1 milliliter of insecticide in the vial, and then check them, roll them, make sure the interior surface is covered with the insecticide, and then empty them and let them dry. And then we load them with 10 flies, 5 of each sex. And after 6 or 8 hours, depending on the insecticide we're testing, we assess mortality. This is the work that was done by Brian Gress, the former postdoc in Frank Salem lab. He collected flies from Cambridge in Watsonville in 2017 and did dose response with spinosad and those flies. If you look at the dotted line in here is the curve for susceptible wolf scale population, and then this dashed line is the one for the Watsonville population, and you can see that the field population was able to tolerate 6 to 8 times greater concentrations than the wolf scale susceptible one. He also selected those populations for 5 generations in the lab, and he showed that even the resistant increases after that selection, and this is the solid line that has shifted more. So following that, we also did dose response bioassays with populations that we collected from strawberries. In 2019, 2 populations were sampled, one from Watsonville and one from Santa Maria. And if you so you can see that the LC50 values were 2 to 4 times higher than the LC50 value for the wolf scale susceptible population at that time. And then in 2020, another 4 populations were collected from Watsonville, and there was an increase in the LC50 values after a year, and the numbers or the concentrations were 4 to 9 times greater than the wolf scale in 2020. We also did bioassays with Mustang Max. It's not registered for using strawberries, but we use that as a proxy for other pyrethroids like Brigade or Danitol that are commonly used for other pests in strawberries. And so in 2019, again, those 2 sites were surveyed for resistance to Mustang, and the resistant, so the LC50s again was 7 to 9 times greater than what we see for the wolf scale population. And in 2020, the populations collected from Watsonville, they were even more resistant. Again, the LC50 values were something from 19 to 36 times greater than the LC50 value for the wolf scale, which is our susceptible. And these rates that are needed to kill 50 or 90% of the population is much higher than the maximum label rate for this insecticide at 200 per 100 gallon per acre. Since we saw resistant to Mustang Max, we also did dose response with bifenthrin to see if there is any cross resistance between these 2 pyrethroids. And as we expected, the flies were also highly resistant to Brigade. And the resistant ratios were even higher, range from 16 to 48 per Brigade. With the populations that we collected recently from Oxnard, we did discriminating dose bioassays using the 3 different insecticides that we have been testing. So the wolf scale is here again is our susceptible population that gives more than around 90-98% mortality, and the S1 and S2 are field populations. And you can see we see some levels of survival with spinosad at this rate, and also for melatonin it was 80%, so it's pretty high, and for Mustang Max, this is the mortality, so the survival was almost all of them survived. So we know that spotted wing Drosophila has become resistant to all of these different insecticide classes. So to summarize this work, we know that Drosophila larvae are present in both ripe and overripe or fresh market and processing fruits from all locations, and 0 to 100% of the larvae, depending on the location or site, where spotted wing Drosophila. We know that spotted wing Drosophila infestation makes fruit more susceptible to other species, and resistance to spinosad, malodin, and pyrethroids is already present in commercial fields in California, and this susceptibility is declining from year to year, and it will likely decline more with continued exposure to this insecticide. So our future research is to assess the stability of resistance in these populations that have been identified as resistant. We want to know if we remove pressure, insecticide pressure from these populations, how long does it take for them to become susceptible again. We are also evaluating the effects of some synergies with spinosad and pyrethroids, and also working to determine the precise levels of resistance to malodin in adult flies. We've done some work with the larvae, but don't know the levels for the adults. We are working on identifying mechanisms of resistance in these flies, and also working with our collaborators to develop and implement alternatives to chemical control, like behavioral control. With that, I would like to thank you, my PIs, and also our collaborators that help us to collect the samples from all regions in California in our lab. And yeah, and I think we can see the poll questions that we have.
Yes, I just launched them. There are 2. Please go ahead and fill them out. We'll give everyone about a minute and a half to do so. And then if you can just go over the answers. So I see a couple people are starting to fill it out.
Sure. So the first question was, insecticide resistance has been detected in spotted wing Drosophila to which of the following classes of insecticides? So the answer was all of the above. We have seen resistance to all these insecticide classes so far. And it seems like 77% got it right. The second question, spotted wing Drosophila are more likely to infest firm strawberry fruit than are other Drosophila species, and the answer was true, and 80%.
Okay, thank you so much for participating, Fatima. And I don't see any questions in chat, but I'll have a chance. Since I have an opportunity, I can ask you a quick question. Did you see the same levels of resistance in organic fields versus conventional fields, or hasn't been considered at all?
Yes. So I think 2 of the fields that we sampled are organic fields. I didn't mention it here, and 2 are conventional fields. So, yeah, and that's for spinosad.
And so the resistance was the same level in organic and conventional, or was it different?
Can't remember it, but maybe Frank can help with that.
That's okay. We can look at it later, but I was just curious.
Okay.
Mhm.
Yeah.
Thank you so much for joining us, and it was a really informative presentation. I appreciate that. And
Thank you.
at this point, for all the participants, let's have about 10 minutes break and come back here at 9:45. It might be 7-minute break. At 9:45, we'll begin the program again. So let's have a little stretch and be back at 9:45.
All right, Oleg, would you like to get started?
Sure, yes. Well, hope everybody had a chance to refresh, and we'll continue with the program. And we'll have Andrea Biscaro, my fellow farm advisor here in Ventura, who will talk about biostimulants in strawberries. Andrea works with the irrigation nutrient management, and this was an interesting topic that came around. There are a lot of products out there, and it's really good to have a thorough assessment. So I'm going to turn it over to Andrea to discuss his findings in the strawberry project that he had. Andrea?
Sounds good. Thank you, Oleg. Yeah. Can you hear me?
The sound is a little bit off, but we can.
Yeah, why don't you turn off your camera since the sound is coming in a little choppy? That might help with the connection.
What
Perfect.
about now?
Yes, we can hear you.
So great. Thank you. Thank you, Oleg, for inviting me to share the results of our study here at this meeting. So my name is Andrea Biscotto. I'm Irrigation Water Resources Advisor based in Ventura County. And we'll be talking about this project where we assessed the impact of different biostimulants in strawberry production. So I'd like to start with acknowledging the co-PI, Patrick Brown. He's a distinguished professor from UC Davis. He was very instrumental to define the specific methods and approach for this project, and also Chris Greer and Oleg Dogovich. So I'll give a brief introduction on why we're doing this. So we have a lot of data to share. So I estimated that about 60% of strawberry growers use biostimulants, and the most common products are the humic substance. So the definition by the European Union or the European regulation for biostimulants is that it stimulates plant nutrition processes independently of the product's nutrient content, right? So the use of biostimulants can add a significant cost to the production of strawberries. However, there's limited research from local universities, and the research findings are often contradictory. Sometimes you have yield increases, sometimes or often nothing, and sometimes actually also yield decreases. So we're asking the questions if the biostimulants can help improving strawberry production under the circumstances of this study. So I'm trying to move my slides in here. Somehow that's not working.
Okay.
Okay, so we decided to do this study under a stress condition of limited nitrogen. Why limited nitrogen? I believe that's one of the main factors affecting strawberry production, or abiotic stress affecting strawberry production. And biostimulants usually work on certain stresses conditions. So next slide, please.
Do you see the screen?
Yes, I can. So sorry everyone about that. It must be the internet connection here at the office. So, all right. In the left, we see an example of off-site movement of nitrate going to groundwater, and to the right to surface water. Next. So because of that, the State Water Resource Control Board, as most of you know, created different regions. We're in region 4. There's also region 3 with strict regulations for nitrogen use. Next, please. In Ventura County, we need to report nitrogen use based on these nitrogen management plan worksheets. And the bottom line with that is the regional board is interested in the difference between applied and removed nitrogen. That's most likely going to change next year, or is going to change next year, maybe most likely. Like the next slide, please. The next slide shows the limit table for region 3. So as you can see, in a few years, there will be a limit per acre per year of how much nitrogen can be applied as a difference between applied and removed. Okay, so that means as the years go on, regulations will get tighter and tighter as far as nitrogen use. So we hope that these biostimulants can help with addressing some of that shortening on how much nitrogen we can apply per crop. Next, please. So let's get into the studies. This study was conducted in a commercial field under conventional production of strawberries. It's typical setup for Oxnard, 6.4-inch bed, 3 tapes. The cultivar was Fronteras. We didn't apply any pre-plant fertilizer in the entire block. It was a sand-loam soil with 1.3% of organic matter. We only had about 4 inches of precipitation throughout the season. We did 48 harvests throughout the season for this study. There were a total of 16 treatments. 12 of those were applied every 2 weeks through the drip tape, and 4 of those were sprayed every 30 or 60 days. And I'll talk about those in the next couple of slides, please. Okay, so we had a randomized complete block design, replicated 4 times. Each plot was 25 feet long and 1 bed wide. We collected soil leaf blade sample analysis for total nitrogen content, and also we collected nitrogen content from the fruits. We did NDVI image every month. We collected above ground biomass, nitrogen uptake, and soil nitrate at 12 to 24 inches depth at crop termination. So we're not going to be mentioning the name of the participating companies to follow university policies. So this is a trick part of this sort of a project. We're dealing with commercial products. We have a choice of either don't do projects like this or do the projects like this and retract the name of the companies. But what's important is that you know what the active ingredient of each of one of these products are. Next, please.
You don't.
Okay, so yeah, that's good. Thank you. Those are the 16 treatments of the study. As you can see, 1 and 2 are the controls, meaning all they received was the mineral fertilizer applied as AN20. So control A had 100% of the expected nitrogen uptake rate, and control B 60%. And I'll talk in the next few slides about what is the expected nitrogen uptake rate. That's based on previous studies we did. And then we have the different colors for all the other treatments. They mean that they're the same product or products from the same company. Okay, 3, 4, and 5 is a nitrogen fixing bacteria applied with control A, 100% of the expected nitrogen uptake, 60% and 20%. And then we have treatments numbers 6 and 7. That's a humic substance applied with 100 and also with 60% of the expected uptake rate. And then you have treatment number 8 is a typical humic substance used in the region, applied with 60% of the nitrogen at a typical rate of 0.5 gallons per acre per week. That's a rate for the biostimulant. And then we have this other product, treatment number 9 is another type of nitrogen fixing bacteria applied with 80% of expected nitrogen uptake rate. Treatment number 10 was applied with 100% of nitrogen, and that's a very, very typical seaweed extract product used in this region with the typical rate applied. Treatment number 11 and 12 is a humic substance applied with 100 and also with 60% of the nitrogen. And then treatments 13 through 16, they're all sprayed. So 13 and 14, they received 100% of the nitrogen. 15 and 16 received 60% of the nitrogen. 13 and 15, they were monthly sprayed of amino acid and peptides. 14 and 16, they were sprayed every 2 months. And those are sprays with plant growth regulators, amino acid, vitamins, and 2 other compounds. So the expected nitrogen uptake rate was 4 and 9 pounds of nitrogen per acre per week. 4 for October through March, and 9 from March to June. Next, please. And I say October to March, so that's for this year. We had late production. A typical year would be changing these rates in February. So this year was at a little colder than usual. Next, okay, so this is basically showing how we define these rates. Let's focus on, can you go back one slide, please? Okay, so the table on the bottom right shows what the rates were for 180, 60, and 20% of the nitrogen. But again, that's based on another study where we quantify nitrogen uptake for 4 planted strawberries. Next slide, please. This is a picture of the study showing where we actually, this white PVC pipes is the manifold where we apply the treatments, and you see the plots in front of it. Next slide, please. Okay, this slide is showing soil moisture, basically to say that water stress was not a factor for this study. We stayed most of the time between 8 and 10 centibars for 6 and 12 inches depth. That's ideal for strawberry production. Next. Next slide, please. That's showing the irrigation, basically 2.4 acre feet of total water applied. Very typical for the region. Next slide, please. This is showing cumulative marketable yield. Basically, this inflection in the curve seen around May 5th, that usually happens before maybe a month or a month and a half before. Next slide, please. So we delayed the change of the rates of the application from October to March through from October to February to October to March. That's the early rate for the season. This is showing in the red line that the average temperature for that season was below the 10-year average in the black line for maximum, average, and minimum temperature. Next slide, please. Okay, this is the main slide with the results for this study showing marketable yields in pounds per acre in the y-axis. And then you see in the x-axis, you see all the treatments of these studies. They are grouped to the left, all in blue, means they all received 100% of the expected nitrogen uptake rate. Number one is control, and all other treatments have a p-value on top of each one of them. So that means that p-value was a two-way analysis between that treatment and the control treatment. So the lower the p-value, the higher the chance that those differences seen between each one of those treatments and the control is statistically significant. So you can see treatment number 3 is the one of the treatments with the lowest p-values, and that product is seems to be the best performing product, very consistent. As you can see, treatment number 4 to the right, when you see the green treatments, all the ones that received 60% of the nitrogen as a mineral fertilizer. Treatment number 4 also had a relatively low p-value. So in agriculture, often you see people going from 0.05 to 0.1 as a p-value showing that the differences are significant. That's because there's quite a bit of variability, or it's hard to control too many variables in agriculture. So every other difference was not close to be statistically significant. But you can see there is a trend looking from the left to the right of decreasing yield with decreasing nitrogen application. Next slide, please. Okay, this is early marketable yield from January to March. You see there are some differences and maybe some trends that we observed in the total marketable yield for the whole season, but none of these differences were statistically significant. Next slide, please. This is cool rate. So there are no statistical difference among treatments, but when you group the treatments within their control type, meaning how much mineral fertilizer they received, you can see there is it's statistically different. Basically, treatments that received 100% nitrogen produce cool rate around 18%, where the treatments that received the 60% of the nitrogen produced cool rate about 23, 24%. That was statistically significant when analyzed comparing those two groups. Next slide, please. Next slide will show a different way to look at the same information. Basically, by on the x-axis, you're increasing cool rate with decreasing nitrogen fertilization, meaning at these rates that we looked, which were quite tight rates, they were based on uptakes, there's really not much margin for error. When you decrease the amount of fertilizer, you decrease the cool rate. You increase the cool rate, sorry. Next slide, please. This is NDVI, or Normalized Difference Vegetation Index. It is a measure to look at the plant vigor and greenness of the crop. Next slide, please. I'd like also to ask Oleg. Oleg, in case I know I'm going over time here, so if there's any time you want to wrap this up, just let me know and I'll get to the conclusions.
Okay, I got a few minutes.
Okay, thank you. So on the x-axis, you see the NDVI, and this is a correlation for the average NDVI value at the end of the season compared to yield in the y-axis, and you see a good correlation, meaning the higher the yield, the higher the NDVI. Next slide, please. This is above ground vegetative dry biomass in grams per plant. Next slide. This is showing a trend that's similar to yield. And the next slide will show also a good correlation of dry above ground biomass with yield. Basically, this is the fourth replicated study that we have data with bigger plants, higher yields. This is 2 years. We have 2 years of data with Venturas. We have 1 year with one cultivar, proprietary cultivar, another year with another proprietary cultivar. Never a negative correlation, meaning increasing above ground biomass, decreasing yields. So next slide, please. I'm going to ask you to basically blast it through the next few slides. So we collected soil mineral nitrogen at 3 different stages. Also, we collected the leaf blade nitrogen, and we didn't see any surprise there, except for the leaf blades. We didn't see much differences, and that can be explained by the fact that we're fertigating every other week, and we're collecting the samples in the week we're not fertigating, meaning a week after the nitrogen application was made. So if you're relying on leaf blade nitrogens to assess nitrogen content or deficiency at your crop, you may miss that, because if your sampling happens right after a fertigation event, depending on the cultivar, in my opinion, maybe that level will go up and it won't show the deficiencies. Maybe the best way to sample that would be just before you apply the fertilizer. Can you go past the next few slides, please? Again, yes, one more, please. 2 more, please. One more. Yeah, cost analysis. So basically what we see here, this is the last slide. And what we see treatments, and then how much nitrogen they receive. The fruit sale is in dollars, and everything else, all the other columns to the right are in dollars. Meaning fruit sale is the weekly yield times the weekly price of the strawberry per treatment. And then you have the cost of the end-to-end is about 86 cents per pound of nitrogen. And then see the biostimulant cost in highlighted in yellow, you see a product that was the best performing product, treatments number 3 and 4. That product is still not available in the market, but they estimate the cost to be similar to the cost of a nitrogen fertilizer. So, which is yet to see. NA stands for not available. Costs are not provided by the companies, or they're not yet marked commercially available. Highlighted in gray on that same BS cost column is the cost of the other products that the price were provided. As you can see in the last column, we're compared the treatments, each treatment with control A. And as you can see, most of these treatments, you end up losing money if you reduced your nitrogen fertilization, expecting that the biostimulants will help, except for treatment number 4 and treatment number 4. That's the only exception. Okay, so next slide, please. Next slide, please. Okay, so in summary, we had some differences, and treatment number 3 presented was yield for treatment number 3, which is the nitrogen fixing bacteria, and apply and received 100% of the nitrogen. That the yield for that treatment was greater than all the other treatments, and that was 6.1% greater. The return of investment is mostly negative and varies with product, but mostly with the nitrogen rate. Next slide, please. And I'm running out of time, so I'm rushing through the conclusions. So the AVI values showed correlation with good correlation with yield and above ground biomass. The core rates were significantly affected by fertilizer rates, but mostly not by the biostimulants. So we have some pending data. We'll be glad to share in the future. We're waiting for laboratory results. I don't know, I don't think we have time for questions, and I apologize for the technical difficulties in here today, but I'm available. Next slide, please. I'm available to answer any questions there are. Just please give me a call or shoot me an email. I'll be glad to talk to you more about these results.
Andre, thank you. There are a couple of questions in chat. Let's take a minute to, we've been behind because of difficulties, but let's go ahead and answer those questions while we're all still here.
Sure. So there's a question saying, above ground biomass include fruits or just vegetation? Just vegetation. Good question, Paco. So we removed all those fruits, and we measured the basically leaves and the stem, the crowns. The other question is, Andre, did you measure fruit size, and were there any differences between nitrogen application rates of the biostimulant? So I measured fruit size in a previous study. In a previous study where we also changed nitrogen rates widely, and we didn't see any differences in fruit size. So fruit size is very laborious. You have to count every fruit for every plot for every harvest. So that takes a lot of time. And since I didn't see any differences in the past, we didn't measure that. I wouldn't expect fruit size to be affected by the treatments. What type of coals did you see to increase with lower nitrogen rates? So basically, that's the marketable selection by the harvest crew. It was a commercial harvest crew. They would select. It was mostly some fruit size and some deformity. So yes, so that's basically what the commercial crew did.
Okay, thank you very much, Andre, for stepping in and going through this and answering the questions. And we'll look forward to more information from your trials.
Sounds good. Thank you all. Thanks, everyone.
And we will move forward with the next. Oh, actually, we have to do the. Do we do the?
No, there's no poll
talk?
for this one. No poll for this one.
Okay.
We'll move on.
Sounds good. Well, we're going to switch gears back to our pathology and look at the Botrytis statewide survey, the gray mold in strawberry, and we're being joined by our colleagues from Cal Poly Strawberry Center, Yu Chen Wang and Gerald Holmes, and to give us an update on that work. So go ahead, please.
Thank you, Oleg. Is my presentation working okay?
Yes, looks great.
Okay, so good morning. My name is Yu Chen Wen. I am a MS student at Cal Poly San Luis Obispo, working along with my advisor, Dr. Gerald Holmes and Dr. Shashika Hewamitharena. My topic today is statewide survey of Botrytis fura in strawberry. So I'm gonna start by talking about the Botrytis disease life cycle in strawberry. Botrytis can survive overwinter in plant debris as mycelia, or the dense masses of mycelia form a hard black ring structure called sclerosia. Botrytis can survive through harsh environment with that. And here's there's a picture of Botrytis sclerosia on a dead tissue. It is less than 1 millimeter wide, so you don't need a microscope to see that. And in springtime, the sclerosia germinate and produce large number of spores, also known as conidia. And the spores are easily detached from the conidia spores, and it can be carried by air currents, splashing water, or other physical activity. Strawberry flowers are very susceptible to botrytis. The spore land on the floral part and germinate. It may stay dormant, but when the fruit is getting mature, the brown lesions start developing under the calyx, where the calyx keep a good moisture. Also, botrytis can grow with the detached disease fruit on that as well. And since botrytis has a wide range of growing temperature, in the cold storage, it is continue growing. So that's the botrytis we see in the clamshell. In the field, the botrytis continue growing and produce more spores and start another epidemic. So I include here is the disease triangle. A disease is occurring with the presence of susceptible host, the pathogen, and a conducive environment. Here, I would like to mention that the conducive environment for botrytis is the optimal temperature with 62 to 77 degrees Fahrenheit and prolonged free moisture, which is more than 30 hours. That could be from rain, from fog, from dew, or from irrigation. So these two factors are very important environment conditions for botrytis disease development. With that, I would like to introduce the Strawberry Advisory System to you. It is a web-based system that supports fungicide application decisions. It is developed by researcher from University of Florida. Dr. Natalia Perez, she's a plant pathologist over there, and she shared this information with us. And you can see that the Strawberry Advisory System is widely used in the East Coast for botrytis with the bee and anthracnose diseases. So the color is referring to the disease risk. So if you see here for the botrytis disease, the risk factor goes from 0 to 1. When it is under 0.5, it is low risk, and it is displayed green. When it is moderate risk, when it is 0.5 to 0.7, it is yellow, and it is displayed yellow here, here. And when it is high risk, when it is risk factor is 0.7 to 1, it is displayed red. So considering the risk factor on that day and a grower's fungicide application condition and the current bloom condition, the system made recommendation on fungicide application. Here in California, we conducted a statewide botrytis fruit survey in strawberry in 2020. So we would like to understand the disease prevalence and its relationship with weather conditions. We collected strawberry from the 3 mainstream of strawberry production district, Oxnard, Santa Maria, and Salinas. So there were a total of 12 ranches were included in this study, and 7 varieties, and some conventional farms and some organic farms. I think there are 9 conventional and 3 organic farms. And we collect them for the whole season for each ranch. So how do we do that? So with a huge support from the Strawberry Commission team, we collected 2 trays of the marketable fruit from the field and shipped them to Cal Poly, and we stored them at 34 degrees Fahrenheit for 7 days. And then we take them out. We did a Botrytis flora assessment. So when we did that, we removed the calyx to better see the fruit and the symptoms. And here is a good example of early symptom with small lesion that just started under the calyx. And nearby the field, we also set a weather station to monitor weather parameters such as temperature, rainfall, leaf wetness over time. Yeah, here's the early symptom of botrytis. So what did we find? So you can see that the average botrytis disease incidence here in this blue bar. So we pull all the sampling from each district. So we see Oxynar, the average botrytis incidence is about 1.5%, and in Salinas it is about 0.5%, and it is about 1% in Santa Maria. They are all below 2%, which is the standard for US #1 grade strawberry. And here we see the 4 ranges we have in Oxynar. So here we show that the average botrytis incidence in black and the average risk factor in blue. So we see that range B and range C, they have a low botrytis incidence of 0.31% and 0.72%. And their risk factor are low as well, with 0.27 and 0.19 here. And seeing the next one, range A, it has a higher botrytis incidence of 2.24%. And this range is with the variety BG 1975. And the risk factor is not so high, just 0.29. And seeing the range D, which is an organic farm, and we can see that with high risk factor 0.57, it is a more conducive environment for botrytis, and that contribute to 2.54%, the high botrytis incidence. And in Santa Maria, we have 6 ranches. Let's see the 3 ranches here, range F, range H, and range E. They were harvested at the same time, early in the spring. So we see that they have the same risk factor level around 0.18 and 0.16. Just F and H, they have lower incidence, and E with the variety BG6.3024, it get higher incidence of 1.07%. And the other 3 ranges, N and O and P, we see that the incidence is slightly higher, especially P. We see that the risk factor is 0.52. So it's also a conducive environment, and that lead to higher botrytis incidence of 2.666%. So next, in Santa Maria, we have 2 ranches. They are very close to each other with the same variety. Just one is organic and the other is conventional. And the conventional is last longer, 1.5 months longer than the organic. So we can see that the conventional has a little bit higher of the risk factor level, but it is still, and it has a little bit higher of Botrytis incident, but they are all very low. So with that, we conducted further statistical analysis to see the effect of variety, conventional or organic practices, month, rainfall, risk factor, and their relationship on Botrytis incidence. And the yellow here highlight the factor that is significant related to Botrytis incidence. So here we see the graph of risk factor and Botrytis incidence. It is significant. The risk factor is significant to the Botrytis incident, and we see that they are in a positive association. That means the risk factor increase while the Botrytis incident increase as well, and that occur in both organic and conventional practices. And this is the first time we see the risk factor applying to California. So we see a positive result. So next is the relationship of rainfall and Botrytis incident. So rainfall is also significant to Botrytis incident. The rainfall here means the accumulated rainfall amount 7 days before the sampling days. Here we see the Botrytis incidence and the rainfall has a positive association as well. But interestingly is that the organic farm, the rate increasing is more than the rate of increasing in conventional farm. So we know that there's organic fungicides available in the market, but they don't show consistent efficacy against Botrytis disease. So this is telling us maybe rainfall is a significant effect on the efficacy of those organic fungicides. So next, we see that variety is also significant to Botrytis disease. Here we have all 7 varieties. BG6 3024 has higher incidence than Mariposa and Fronteras. And the rest variety, BG 1975, Monterey, PS 9271, and Marcus, they are in the middle. And next, very interesting, we look at the conventional practices and the organic practices. There's no differences between these two practices, and they are at the same level, around 1% of Botrytis disease level. So with that, I would like to summarize some observations we found in this study. So first of all, the post-harvest Botrytis level were very low, mostly. It's less than 2% in average. And second, rainfall, risk factor, and variety, they showed significant effect on Botrytis level. We also see the coastal effects on the risk factor and Botrytis level. And third, organic and conventional practices showed the same level of Botrytis disease. Knowing that most of the organic fungicides are not so effective against Botrytis disease, we found that maybe there is a potential that we can reduce the fungicide application for the conventional practices. But of course, we need to see this for more years, especially when the conditions are favorable for the disease. With that, I would like to thank the strawberry growers who collaborate with us, the strawberry commission team who help us facilitate the research, coming with the grower, and the Cal Poly Strawberry Plant Pathology Lab for providing technical assistance, and the Cal Poly Statistics Department for provide statistical assistance. And so that's all. Thank you for your attention. And so next, I think we have the poll question, right? So the first one, which of the following environments is required for Botrytis fura disease infection? Prolonged wetness, more than 30 hours and temperature above 80 degrees Fahrenheit. Prolonged wetness, more than 30 hours and temperature between 62 to 77 degrees Fahrenheit. High relative humidity, more than 80% and temperature between 62 to 77 degrees Fahrenheit. And the next one, what is the primary function of sclerosia in the Botrytis disease cycle? Infection, reproduction, or survival? And I think we need some time for the, okay, for the first one, the answer is the second one, prolonged wetness more than 30 hours and temperature between 62 to 77 degrees Fahrenheit. And I think most of you answered this question, right? And the second question, also most people answer it right. The function of sclerosia is for survival. So with that, do we have time for questions? And anybody have questions?
I don't see any questions in the question and answer box, but I have a quick one for you. It's when you looked at cultivars varieties of strawberries and you compare them and give them different ratings, what was it approximately the same size of canopy or the same space that they occupy, where some of the plants bigger than others?
That could be,
varieties?
Yeah, that could be a issue because if the canopy is bigger, then the plants are not gonna be dry faster, right? So that you bring out a very good point that the variety might affect the canopy size, and also the farm practices, they might affect the plant canopy, and also the calyx position, is that oppressed or is that reflex? Those might affect that as well. But we don't have that information. It's a wide survey, so the grower collaborate with us. We only know the variety information so far. If we need to know that, we may need to conduct a variety trial on that.
All right, and thank you so much. Thank you for joining us and discussing your work.
Thank you, Oleg.
We will continue with the next talk, who's also talking about one of the pathogens we haven't talked much about yet. It's another presentation from Cal Poly Strawberry Center. I believe Jack Koster is going to be presenting, and he worked with Shashika Tarana on this project. And I think I'll pass it over to Jack to present. It's about Verticillium control in strawberry.
Good morning, everyone. Do you hear me well? Jack is going to take over when I introduce him. Thank you, Oleg, and the team for inviting us. I'm going to talk to you today about an integrated approach to control Verticillium wilt of strawberries. I'm sure you all have heard of Verticillium wilt. This disease is caused by a pathogen named Verticillium dahliae, and you can see the disease symptoms here. And when you culture the pathogen, you can see the signs of microsclerosia on the media. At our strawberry diagnostic service at Cal Poly, we receive a lot of Verticillium wilt positive samples from the northern districts, Salinas and Watsonville, compared to the Santa Maria and southern districts. And This pathogen has a very wide host range, including some of the rotational crops. Over here in this table, you can see the crop and the threshold of colony forming units, or the propagules of the pathogen in soil. And you can see that strawberries has a very low threshold that would show the symptoms of the disease compared to a rotational crop like lettuce. So it's not favorable for a strawberry grower to rotate with lettuce under these circumstances. Now, let's talk about disease management. The disease management is mainly focused on breaking the pathogen life cycle. Over here, you can see the pathogen life cycle, and this fungus produces these survival structures called Macrosclerosia, which can last in soil for about 14 years without a host. And when you have the new plants planted into the soil, these Macrosclerosia germinate and infect the strawberry roots, and it colonizes the plant and reproduces and make many Macrosclerosia in that plant. And when you disc it into the soil, you're basically burying that inoculum back into the soil for the next crop. The disease management is focused on tackling these stages of the life cycle. And you heard from Glenn about cultivar resistance. And when you fumigate the soil, you're basically trying to reduce the Macrosclerosia level in soil, and to some extent, Macrosclerosia in crowns and root, the residues of the crowns and roots. And crop rotation, we talked about that it might not be a suitable option if you're rotating with the host of this pathogen. And recently, crop termination has been introduced to control soil-borne diseases. Crop termination targets the Macrosclerosia production in the crown and roots of the plant. Crop termination is a method that uses plants' ability to uptake a fumigant that has herbicidal and fungicidal properties and kill the fungus within the plant and prevent it from producing those Macrosclerosia after the last harvest of the crop. And it is different from bed fumigation because in bed fumigation, you won't have the plants. And there have been previous studies that looked at crop termination, for an example, for Macrophamena, Fusarium, and to some extent to Verticillium Wilt. However, we still have a lot of knowledge gaps. For an example, is crop termination itself adequate to reduce the inoculum level to below the threshold for strawberries? Is there a difference between registered fumigant products for crop termination? What is the most suitable stage of the crop to terminate? With that, I'm going to ask you a question. If you have terminated your crop in the past 5 years to control any soil-borne pathogen, please answer yes or no. Okay, so most of you have not used the technique, and it's good to know there's a potential to use this technique. And with that, I'm going to turn the presentation to Jack Coaster, one of our Cal Poly Master students at the Horticulture and Crop Science Department and Strawberry Center. Jack?
Okay, thank you very much, Dr. Hewa Vitharana, and good morning, everyone. I'm going to take over and talk about our objectives for this experiment. Overall, we want to develop new and enhanced soil-borne disease management practices for the California strawberry industry. Further, we'd like to determine the effectiveness of the sequential application of crop termination and bed fumigation in their decreasing Verticillium dahliae survival. Further, we would like to evaluate the efficacy of the integration of resistant cultivars with the chemical methods in order to lower the Verticillium Wilt inoculum levels. So next, let's go ahead and talk about our experimental design. Here, what you are seeing on the left side is our Field 25 Block 6 here at the Cal Poly campus. I wanted to show one replicate in order to describe the experimental design easily. So as you can see here, we have a randomized split plot design, and our whole plots are separated by fumigant type. So you can see denoted here, KPAM, VPAM, and trichlor. And our subplots, 3 per bed, are by the method of application. So for instance, we have bed fumigation, we have the sequential application of crop termination and bed fumigation, and then we also have crop termination by themselves. So overall, we had 12 industry standard beds. They were 90 meters in length, and there was roughly 930 plants per bed. There were 4 rows of crop in each bed and 3 lines of drip. Overall, we had 9 treatment beds and 3 controls. You can see the control beds denoted here by the brown. So now that you've seen it as on a schematic scale, let's look at it from an overhead shot. So actually what you're seeing here is our crop termination plot here at Cal Poly. Outlined in yellow here are our subplot barriers. So as I previously mentioned, we had 3 different subplots that had different methods of application. In here, you can see in the blue, we have our control bed. And in the black, you can actually see examples of the terminated subplots. So you can see significant differences, for example, between this plot that was terminated and this plot that was not. You can still see some relative plant death here, and as well as in the control bed. So when we're talking about the terminated subplots and the crop termination itself, how exactly did we do that? Well, we were looking at our crops, and when they reached about 5% mortality and about 45% symptomatic expression, we went ahead and did our termination application. That ended up being roughly 240 days after transplant. We used KPAM and VPAM for our crop termination, as these fumigants are labeled for such application. You can see their application rates here. And for bed fumigation, we will be utilizing trichlor since it is labeled for bed fumigation, and this will be taking place next month in October. So next, let's talk about how exactly we went about collecting our data. First off, we wanted to see how the crop was injured by this termination application. So we went ahead and visually assessed each plant in the crop, and we scaled it from 0 to 4. So on the left side, you can see our scale, for 0 being 0% mortality and 4 being 100% mortality. And over here, we have a really good picture of the wide range of crop injury. You can see it from 25%, where we really don't see a lot of wilting. Half the plant parts are not necrotic. And then you can see up here in 100%, we have complete wilting and all plant parts necrotic. Up here, you can see that we still have green in the center, and the apical meristem is still green as well. That was classified as 75%. Now, next, we also wanted to evaluate how we can quantify the pathogen density in the soil. So what we did was we collected the soil from our experimental plots and pooled them and let them air dry on a lab counter for about 2 weeks. And once that was done, we used a mortar and pestle and grinded it into a fine dust, where it was then sieved in a 150 micron sieve. We went ahead and weighed these samples out to 0.1 grams and used an Anderson sampler to plate the soil onto a selective media NP10. Now, these plates were left alone in the dark for about 15 days before we went back and counted or quantified the amount of Verticillium dahliae CFUs. And this is an example of a Verticillium dahliae CFU, and you can see these black overwintering structures. So next, we also wanted to evaluate, well, how did the pathogen survive in the crop after we terminated it? So what we did was we collected 10 plants from the middle 15 meters of each terminated plot, and we processed them and plated them on a selective media NP10. And what we wanted to see was whether or not the pathogen survived in both the crown tissue and in the petiole tissue. So you can see here, we collected plants with a wide range of injury scales, and we brought them back into the lab and plated them, and then analyzed them for the presence of verticillium. You can see here the petiole infection of verticillium, and you can see here the crown infection of verticillium. So now that I've reviewed the materials and methods for this project, let's go ahead and look at some results that we have for you all. I wanted to show you all this overhead drone shot that we have here, and it just shows you the progression of the crop termination as the days progressed. You can see here, this was done 2 days before we terminated our crop. You can see substantial wilting and necrotic tissue in some plants. You can see some plants are missing. And then about 6, 20, and 34 days after application, you can start to see the progression of plant decline. Over here, we see some drop off and some wilting, loss of vigor. And then as we move into 20 days after, we start to see some real necrotic and real dead tissue. So when we're looking at crop injury at a percentage-wide scale, let's go ahead and look at it at 7 and 14 days after fumigation. And we wanted to do this because we wanted to see if there was a difference at 7 and 14 days. Time is money. So is there a difference in waiting? So at 7 days after fumigation, we found that the overall injury was roughly 77%, with KPAM being very similar to each other in terms of delegation. We actually got close to 90% in our terminated crop. What we did see, though, at 14 days was a significant difference between KPAM and VPAM in terms of terminating the crop. So KPAM gave a significantly higher percentage of crop injury compared to is roughly 12 to 13 CFUs per gram of soil. Now, after we terminated the crop, we see down here that our KPAM and VPAM inoculum densities are significantly lower. So with KPAM, we had roughly just above 5 CFUs per gram of soil. And with VPAM, we had just about 4 CFUs per gram of soil. There is no significant difference between the two fumigants in terms of decreasing pathogen density, but both of these were significantly different from the control. And what we see here, as Dr. Hewa-Vitharana mentioned, is we have that minimum strawberry threshold of 1.9 CFUs per. Okay, so let's go ahead and pick back up here. Before we did any application, our pre-plant CFU was around 7 or 8 CFUs per gram. After the crop termination, our control was roughly 15. And at the post-crop termination for KPAM and VPAM, we were hovering around 5. Now, for our subplots that are getting bed fumigated, that had a roughly a 12 CFU per gram inoculum density in the soil. And this is a good thing to see between the control and the pre-bed fumigation inoculum densities, for we want to see if what bed fumigation will do to such a high level. So next, let's go ahead and summarize everything together and talk about pathogen survival in the crop in terms of odds. So what you're seeing here is a log odds ratio figure. And this is what it's showing is the verticillium survival in the terminated crop in terms of an odds ratio. So let's start here with KPAM versus the control. You're roughly 4 times less likely to have verticillium survive in your terminated crop than it was in the control crop. We see roughly a similar odds ratio for VPAM. You're roughly 4 times less likely to have verticillium survive in your terminated crop versus that was a control and not terminated. And let's also look at time. You can see here that at 14 days, you're almost 2.5 times less likely to have verticillium survive in your terminated crop compared to that at 7 days after fumigation. So let's go ahead and bring everything together and give one final take-home message. In terms of crop injury, we saw a substantial amount of crop injury from our termination, with 14 days after fumigation approaching almost 90%. And at this 14 days after fumigation, we did see KPAM performing significantly better than VPAM in terms of delivering injury to the crop. Next, in terms of soil inoculation density, inoculum density, KPAM and VPAM significantly reduced the pathogen density in the soil compared to that of the control plots. The fumigants, although, were not significantly different in terms of controlling inoculum density in the soil. And after the crop termination, we still are above that CFU threshold for strawberry. Finally, in terms of pathogen survival, I mentioned that you're roughly 4 times less likely to have verticillium survive in crops terminated than those that were not. And you're more likely to have the pathogen survive in the petiole compared to that of the crown. Now, the log odds of verticillium surviving is roughly 2 times lower at 14 days after fumigation compared to that of 7 days after fumigation. So with that, I'd like to now turn to our survey questions and allow you to answer them for us. To start, we'd like you to identify which correctly describes crop termination. Is it the process of applying an herbicide to kill the strawberry plants? Is it the process of delivering a fumigant via drip in order to kill pathogens in the soil? Is it the process of delivering a fumigant via drip in order to terminate or kill the crop to eradicate pathogen survival structures? Or is it D, the process of using carbon source and irrigation to create anaerobic conditions to control soil-borne diseases? The second question is whether or not KPAM was significantly better than VPAM in terms of decreasing pathogen density in plant tissue. So we will give you guys time to answer these questions. Great, so let's go ahead and talk about the first question. Around 80% of you got the question correct, and crop termination is the process of delivering a fumigant via drip to terminate or kill the crop in order to eradicate the pathogen survival structures in plant tissue after your last harvest. No one selected answer D, which is good to see. The process by using a carbon source and irrigation to create anaerobic conditions. That's anaerobic soil disinfestation. So the next question was a true and false question. KPAM was significantly better than VPAM in terms of decreasing pathogen density in the plant tissue. 56% of you said true and 44% of you said false. This was a little bit of a tricky question. So KPAM was significantly better than VPAM in terms of delivering injury or creating a termination effect. There was no significant difference in terms of decreasing the pathogen density inside the plant tissue itself. So thank you all for participating in that. And our last question before we have you all go is, as a grower, what major concern would you have for doing a sequential application of crop termination and bed fumigation? Let's say your field is heavily infested with a soil-borne pathogen. What would be holding you back from doing this sequential application? Okay, great. So it looks like all of you have identified all of these as a potential concern, and we appreciate the feedback moving forward. So with that, we'd now like to give some acknowledgements. We'd like to thank the Agricultural Research Institute for funding this project, along with the California Strawberry Commission. We'd like to generously thank TriCal and AMVAC for doing our fumigation for us and providing us with such fumigants. And we'd also like to thank our Strawberry Center pathology team, Vivian Longacre, and our undergraduate student assistants. And with that, I'd like to thank you all for listening, and we can entertain any questions that you all may have. Thanks.
Thank you, Shashika and Jack. There's one question in chat. Would you like to answer this? Or the question is, could the crop termination increase Fusarium inoculum in the soil?
So this is not based on our work, but from Oleg, you did some of this work, correct? With
Yes.
With Peter Henry. I don't know whether you look at the soil inoculum level.
Yeah, we did look at both. We did a bird inoculum. We did also infested crowns, and generally it decreases the Fusarium inoculum in soil, especially effective when it's just Sclerosia mixed in soil. And just to point out, we talked about Verticillium. We looked at Macrophamina and Fusarium, but the other targets such as perennial weeds, such as nutsedge. So there's multiple objectives with this technology. So hopefully the costs can be broken down for different other objectives as well. They'll look more palatable.
Yeah. Thank you very much, Oleg.
Thank you so much for joining us today, and appreciate your time and presentations. Now, to wrap it up, we heard this during this meeting, and you all know that insecticide resistance is a big problem. Just to every mode of action, we have resistance both in flies, ligas, and other species of insects. So hopefully there's no resistance to back vacuuming, and it's been continuously focused on improving the back vacuum. And John Lynn's here today to join us with the latest research on the back vacs. So John, he's with Strawberry Commission. He'll been an expert in this subject. Go ahead, John.
Thank you, Oleg. I'll start up right now. So John Lynn, Production Automation Manager, California Strawberry Commission. We're going to talk about two topics today, the Ligas bug vacuum and the 4-row spray rig. Essentially, the bug vacuum is now commercially available. You can see the new bug vacuum in aluminum. We call it the double barrel bug vacuum. This is for 4-row production in the Santa Maria Oxnard region. We also have a single barrel version for the Wasma Salinas So this new version essentially is now commercially available at your local CNN dealership. And we also will be posting details online if you want to self-fabricate. So this is the single barrel bug vacuum located in the Watsonville Salinas area. And again, this one is the double barrel located in the Santa Maria and Oxnard. The main difference is that you're going to see that the new versions, both the single and double, they increase airflow significantly. I'm not sure if these videos are coming through. I try to increase my bandwidth as much as possible, but you can see just before and after the vacuum, you can see significantly more agitation and debris being removed by the new versions. And this has resulted in about a 3.1 times increase in ligas removal from the single barrels, so that's the Watsonville Salinas, and the 2.3 times removal from the double barrels, and that's the southern districts. And again, the main thing is just airflow. It's all about airflow. The components that are different, how do we increase airflow? Essentially from the old versions, it kind of had a funnel design. So we changed that to a tubular design. So you can see in the barrel, the double barrel version or single barrel is now tubular. We also raised the baffles. So the metal plates where the ligas impact and eliminate, those were actually causing a choking of the vacuum, which decreased airflow by like 10 to 15 miles per hour, which is significant. Those are the main things. And then we also try to make this for the southern districts. We know a majority of tractors out there are 90 horsepower. So we try to make this retrofittable to the current tractor. And for the Watsonville Salinas, the single barrels up north, this is a three barrel system you see here. We also have 4 barrels. The image you saw earlier that's commercially available at your local dealership is 4 barrels. But we try to make this fit a 70 horsepower tractor that's commonly found in the Watsonville Salinas area. But again, this is all this design is the same, tubular, increased a little bit of extra room for your baffles and selected motors and hydraulics to increase the power efficiency. So if you want to build it yourself, all the designs are available online at the Cal Poly Strawberry Center's website. So we'll give you all the schematics, all the components, and walk you through it. We'll give you animations on how to assemble all your parts. If you want to look into this a little bit further, we have a journal publication in the International Journal of Fruit Science. We can go through the details. We also, again, as I mentioned, on the Cal Poly Strawberry Center's website, you can take a look at all the components, where to get them, or local dealership if you want to contact your local CNN dealership to purchase one. And if you're interested in kind of an overview of how it works, talking about all the components that were changed, you can go on YouTube and we have it all there as well. Spray rig update. So we have a new spray rig recommendation for the 4-row system. So Santa Maria and Oxnard. So this has been going on for 5+ years now. But essentially, in the first few years of study, we found that the volume does not equal coverage. Coverage depends on rig design, operator, and weather. So in terms of operator, we have a training program which contact the commission and we'll put you in touch with. But we'll be focusing on rig design for today. So the first thing we did is that we created basically an evaluation yardstick. We went out to 25 different growers across the 3 districts, and then we measured how much coverage are they currently producing. This is after calibration. So they first went through one of our calibration training programs, and then we did this analysis. So you can see here from our point of view, probably 50% of the industry could use a recommendation to increase their their overall coverage performance. We want to see it in above, above 50%, above 50 to 60%. But there are quite a bit of high performers. So what you're seeing here in this graph is that this is specifically the highlighted one is rig 1. It was a very high performer, 89%, and it was amongst their neighbors, which are located in blue stripes, like so all their Oxnard neighbors. But this is the distribution is pretty similar in each of the districts. So I wouldn't say any one district is outperforming another. So while we were doing this analysis, we also calibrated, so that we did 25 or so evaluations. We also calibrated something like 80 plus rigs across the industry. And while we were calibrating, we noted specific parameters of the spray rigs. So we saw heights in terms of nozzle to canopy. We saw heights varying from 0 to 24 inches. We had pressures from 50 to 500 PSI, which is these huge ranges. Quantities per bed, 4 rows, we saw 14 to 20 nozzles, 2 rows, 10 to 14 nozzles, disc types, disc core, cone jet, flat fan. So there's a large range, and we were trying to see, could we find a specific one that was the best out of this, and try to standardize the industry more as well, so that we can implement better training programs and for maintenance and calibration. So we evaluated a lot of the typically found configurations in the industry, and we did find that there was one that either performed as good or better than the rest. And this is the 8 nozzles per bed. And then again, this is just for Southern districts, Santa Maria, Oxnard, 8 nozzles per bed, hollow cone, cone jet TXRs, 80036 VK green at 111 PSI. So this was, again, we will try to accommodate growers in their practices. Those numbers are not completely hard. They're a little bit flexible, but we try to get them as close to that as possible. So in terms of the recommended parameters, we recommend flying the nozzle as close to canopy as possible. So 0 to 4 inches. We really find there's a gold star there because we find that that's one of the most significant factors in providing good coverage. And then there's pressure, 111 PSI, quantities, only 8 nozzles are needed. That's typically about half as many nozzles as our conventional grower puts out on their row. And we found that the less nozzles performed as good or better. And the type of nozzle is cone jet. And we can go into that further. You can contact the commission and we can talk it over with you if you have any additional questions. In terms of what we've seen so far is growers, once we come in and we do a calibration and then we give them our recommendation and they adopt it, they typically see, it depends on the grower again, it depends on where the grower started out, but we've seen numbers of 30–40% increases of coverage. So that's good to see. It's good to see that if you have concerns with your rig, please reach out and we will have a recommendation and try to get you up there. We also have online videos and again, tutorials on how to design the 4-row system. So this will walk you through how to properly wire it all up and all the schematics for the manifolds and the components are all available online. If you want to go through additional details, feel free. We have a journal paper in International Journal of Fruit and Science. You can look up all the details there. Again, the Cal Poly Strawberry Center's website. You can find all of this information, components, drawings, animations, etc. online on our website. And then the Cal Poly Strawberry Center, the California Strawberry Commission also has a YouTube page. And you can find additional details overviewing this whole project. So with that, I'd like to go to our first polling question. I'll go over them. Polling questions, true or false. The new single barrel and double barrel bug vacuums have been shown to remove 3.1 times and 2.3 times more ligas respectively than conventional vacuums. And the second one, spraying as close to the canopy as possible is the single biggest factor to achieving increased spray coverage. Again, true or false? Yeah, so it seems like everyone's got the first one right. The second one, I understand there's a definite preference there with respect to each grower. And based on our observations, evaluating the 25 rigs across the industry and the data that we've seen irrespective of rig design. And again, rig design was varied. We went through it, 50 to 500 PSI type stuff. It was varied, but we saw that irrespective of rig design, that having the nozzle as close to the canopy increased your coverage. And the main thing is because the winds, 5 miles per hour is pretty normal, up to 18, even above. You really want to try to get that nozzle as close to the canopy so that the particles can get into the canopy. And once they're in the canopy, they do tend to trap themselves in there. But if you, when you're in open air, those particles can be taken up pretty readily. So that's just something that we'd say, that's just based on our observations, but definitely understand the polling results there. Okay, that's all I have.
Well, thank you very much, John, for joining us. Every tool in our disposal is valuable. We have different challenges in pest management. So whatever we do, whether it's a bug vacuum or improving our sprays, it's going to be instrumental helping us out. At this point, I'd like to also thank all the speakers and everybody staying on time and being patient with our technical difficulties, and our audience to being patient with some of the glitches that we had. We're just a couple minutes behind the schedule. I hope we'll have this kind of meetings and discussions next time in person over lunch in a more enjoyable setting. But I'd like to thank everybody for joining us today, and turn it over to Kelly for some take-home reminders. Is it Kelly? While I do so, I'd like to also thank Kelly and the program unit support for putting this together and organizing it and making sure it runs smoothly. So is there any last moment reminders from Kelly or elsewhere?
Yeah, I just wanted to remind you that you're going to get a test emailed for DPR. You need to put in your DPR number and then answer the questions and pass with 70%. Everyone will get this email. If you do not need DPR credit, you do not need to take the test. Don't worry about it. Our servers have been down pretty much all morning, so don't expect to get the email until the end of the day today. I'm hoping it'll get back up so that I can send you this information, but it'll probably be closer to 5 o'clock when you get it. There's the QR code for a certified crop advisor. Just open the app, take a picture. And if you don't have the app, just email ANR program support at ucanr.edu with your CCA number and I'll get you signed up. And other than that, I'll leave the meeting open for another minute or two to get this information and the scanning done. Otherwise, everyone's good to go. Oh, recordings. I will get the recording processed and emailed to all registered participants within the next 2 weeks. So it takes a couple weeks to get processed. So, and that's it.
Well, that will be up for a minute, like Kelly said, and thanks again for everybody joining in. We'll looking forward to your participation next time.